So students, let's today learn some of the differences between DNA and the RNA, and then we will move on to two very easy differences between two types of DNA, and then a new topic is going to start. So what is the difference between DNA and RNA? Some of the points I have mentioned here. If you can search more on internet, you may learn. So first thing is DNA is present always inside the nucleus as well as inside the organelles, because when we talk about the eukaryotes have chloroplast and mitochondria. So DNA is present in the mitochondria and chloroplasts, as well as present in the nucleus, as well as present as nucleoid, if you remember, in the case of the prokaryotic organism. But when it comes to RNA will have very little, when it comes to RNA is present to a very little amount inside the nucleus. Its main amount is present in the cytoplasm in the form of, that's why if you remember in the last class, the question I gave you was, where are rRNA, tRNA, mRNA formed? So answer is yes, nucleus. But where do they go and function? So answer is cytoplasm. So where do you find the maximum amount of RNA in the eukaryotic organism? Answer would be cytoplasm. Then DNA exists as the genetic material in all the organism, whether prokaryotes or whether eukaryotes, and even some virus also. But when it comes to RNA, only certain viruses can have their RNA as genetic material. Otherwise, RNA does not act like genetic material. I'm repeating again, DNA is genetic material, it's a rule. But there are exceptional organisms where, that means some virus, where RNA also acts like a genetic material. Otherwise, in maximum organism, RNA is not the genetic material, and such viruses which carry RNA as genetic material are called as ribovira or riboviruses. Now the next thing, DNA is general rule once again, just like genetic material, DNA is double-stranded. But there are again certain viruses which may carry single-strand DNA, and such viruses are called as deoxyvira. So please remember, viruses can be ribovira, which carries RNA, or viruses can be deoxyvira, which carries DNA. Now please remember, that means double-stranded DNA is a rule, but virus may have RNA, virus may have DNA, and the DNA of virus can be double-strand or single-strand. In the same manner, RNA is present in some viruses as the double-strand structure. So that means viruses can have single-strand RNA also and double-strand RNA also. That means rule is what, DNA should be double-stranded, RNA should be single-stranded. But exceptions come with the viruses. There are some viruses which may have single-strand DNA and some viruses which have double-strand RNA. Examples I have already mentioned. Example 174, it's a bacteriophage which attacks bacteria. So it's a bacteria pathogen virus. And then the example for single-strand RNA is reovirus. We'll be learning virus quite late, but it is in your syllabus of 11th class. Now then there are millions of nucleotides which join together to form DNA. So it's a very long chain. But when it comes to RNA, it can be as less as somewhere around 70 nucleotides, or it may go up to 10,000, 12,000 nucleotides. So that means DNA is quite long and RNA is comparatively very short. Next difference possible is the DNA carries deoxyribose sugar, RNA carries the ribose sugar, though both are pentose sugar. Second carbon position carries a side group H. Second carbon position, the side group is OH. So ribose sugar has modified into deoxyribose sugar. This confirms that maybe through the evolution, RNA came first and it gave rise to DNA. Now the next is, when it comes to DNA, the nitrogen bases are A, G, C and T, adenine, guanine, cytosine and thymine. But in the case of the RNA, it is adenine, guanine, cytosine and uracil. Uracil came first and uracil modified into 5-methyl uracil, which is also called as thymine. Next one is DNA. Okay, one more thing we can learn, that because T is present here and deoxyribose is present here, that's why DNA is, because DNA is double strand also. So 3 reasons why DNA is stable and DNA is non-reactive. Reason are 3. First reason is because maximum DNA is double strand except some deoxyvira where it is single strand. So double strand means stability. Deoxyribose means non-reactive. Thymine means non-reactive, but opposite, RNA would be non-stable because it is single-stranded, and it is reactive because it carries uracil and it carries ribose sugar. Next difference is unusual bases. Now what is unusual bases? Modification of the bases. We will be learning this topic in the 12th class, because in detail we will learn the structure of the tRNA. So in tRNA you can find these types of bases, nitrogen bases. So there are absence or very few, that means rarely you will find unusual bases or modified bases in the DNA, but RNA carries many, especially tRNA carries many modified or unusual bases. Next is the renaturation. What is renaturation? We have already learned long time back in the protein topic, and I told you it is found also in the DNA and RNA, that when you do the heating and all, then the bonds will break and the DNA will become unstable or RNA becomes unstable. But once the temperature again slowly comes back to normal, then the DNA and RNA will again become normal with their bonds. So denaturation followed by renaturation. So renaturation is very slow in the case of DNA and it is very fast in the case of the RNA.